Today's lesson is the first of 4 in genetics. We are going to review a topic that we have talked about throughout the year, molecular biology. We are also going to review a topic today, a mathematical concept, probability. Throughout this unit, we want to answer this following question. How do we go from DNA to what we look like? In order to do that, we first need to understand that the processes that we have learned about the past several units are connected. We also need to show the relationship between DNA, genes, and different alleles. Finally, we need to practice applying probability concepts to word problems, because a large part of genetics is probability. So first, let's talk about how DNA replication, meiosis, and protein synthesis relate to one another. Remember that the process of DNA replication is the cell copying or duplicating all of its genetic material during the synthesis stage. So during the S stage of interphase. And this S stage of interphase is where replication happens before mitosis, or even before meiosis. And we have to understand the S stage is in preparation for cell division. So the whole reason we go through this DNA replication process in mitosis is so that each of our new cells has an exact replica of the parent cell's DNA. However, when we go through DNA replication in meiosis, there's a little bit of a difference. Basically, we're going to go through the G1S and G2 before meiosis 1 for a couple of reasons. Number one, we want to make sure that when we create our 4 cells at the end, we want to make sure that our 4 cells are haploid in number. So we're going to go through 2 divisions to get 4 haploid cells. Number 2, we want to make sure that as we go through those 2 divisions, that we allow the sex cells that are dividing to have the chance for genetic variation. So we're going to create genetic variation. And we do that in a couple of ways that we talked about last unit. We talked about doing that via crossing over or independent assortment. So crossing over and independent assortment. Remember that independent assortment happens during metaphase. Independent assortment happens during metaphase, and crossing over happens during prophase 1. Now, the whole reason that we have DNA, the whole point to DNA, is so that we can create proteins. Remember that DNA does not create our traits, but the proteins that we make during protein synthesis are going to create the traits that we see. Proteins can be molecules that we can do a lot with, but they can be structural, which makes up us. So proteins might make up our hair, might make up our skin, but those are going to be structural things, are going to be things that we can see. Other things that proteins are made for is to be used as enzymes and more worker molecules, but those will create traits as well. So our DNA does not create our traits, but our proteins do. So how can we relate DNA with chromosomes and genes and alleles and ultimately proteins? Well, each cell, remember, is going to contain DNA in its nucleus, in its castle, and we said that this DNA was the recipe book. And that recipe book was going to have recipes for lots of different types of proteins. And we called those proteins cookies. But sometimes this recipe is so big that we need to divide it into several volumes. And each chromosome we can think of as a volume of this recipe book. So we might have a recipe, a volume of the recipe book that is our main course, one that's our salads, and ones that are desserts, but each recipe is going to be for a separate gene. And those segments on those chromosomes that code for those recipes, or are recipes, these genes are the recipes themselves. Each recipe then is going to be made through protein synthesis. We're going to go to the ribosome, go through protein synthesis, and we're going to make what we called our cookies. And from this point out, I mean, we'll still probably be talking about cookies, but each protein might not be a cookie. It might be a steak or a chicken or hamburger, something that we've made from this recipe. All right. Now, each protein then is going to produce a trait. It's going to provide a new taste for us. All right. Each protein right now we will think of as producing another trait. Now, we all know that we don't look exactly the same, and the reason that is because of the variations of those traits. And each variation to a separate gene is called an allele. So for instance, if we were making cookies, and we can go even more specific and say if we were making chocolate chip cookies, some people don't make them exactly the same as others. And so one person might be making cookies with almonds or with walnuts. We'll use walnuts. I know my mom likes to put walnuts in her chocolate chip cookies. Other people, like me, don't like walnuts in their chocolate chip cookies. So these alleles are going to be different variations of the same gene. They're all going to code for the same trait, but they're going to be just a little bit different. All right, so an example of this would be, for instance, somebody who has brown hair is going to have a different allele of the same gene as somebody who has blonde hair. And which allele we get is largely due to chance. So probability describes the chance of that variability. So let's talk about the probability of a single event. And remember that this probability happens during prophase 1 and crossing over, possibly metaphase 1, when we have independent assortment, or metaphase 2 when we have independent assortment. We can find the probability of a single event by saying the probability is equal to the number of favorable outcomes, or the number of outcomes that we are looking for, over the number of possible outcomes. So if we think of this in terms of a penny, if we had a penny with heads and tails on each side, what is the probability that we would get heads on a single flip of that coin? So we're looking for heads. Probability of heads, well, what are the number of favorable outcomes, or the number of possibilities that we could have heads? Well, we only have one possibility of coming up heads when we flip a single coin. So we put a 1 for the number of favorable outcomes. Well, what are the total number of possible outcomes on that single coin? Well, we have a heads and we have a tails, so that would give us 2 possible outcomes. The probability of getting a heads when we flip a coin is 1/2. When we do these problems, make sure that you start with the equation and then plug your numbers in as you go. That's something I'm going to be looking for, and it's something that you should hold on to as you go throughout the rest of biology and into chemistry, because the problems get more and more complicated. and that will just help you out. Now, what if we had 2 coins? What if we had 2 coins and I wanted the probability of getting a heads and a tails from flipping those 2 coins? Well, that's the probability of 2 single events happening at the same time. So we can figure out 2 or more single events by saying if I want the probability of a single event A and another event B, well, that's basically going to be the probability of A times the probability of B. So in our example, I got 2 coins. I want a heads and a tails on the coin. Basically, I'm looking for the probability of heads and the probability of tails. Well, I've already figured out the probability of heads by taking the number of favorable outcomes over the number of possible outcomes, 1/2. Well, we need to do the same thing with tails. The number of favorable outcomes on 1 penny for tails is 1. The number of possible outcomes is 2. When I multiply those things together, I get 1/4. So the probability of getting a heads and a tails on 2 separate pennies is 1/4. That's a very basic probability problem, or both of those are very basic probability problems, but it'll get you back into the swing of things, and we're going to be doing more and more probability as we go through this unit. So tonight I'd like you to look at these 4 questions and write down the answers. You have these 4 questions in your packet. And the first 2 you're going to have to do a little bit of writing, but it doesn't have to be in complete sentences. Just get the ideas down. Just make sure that you can explain what you're doing with these 2 problems. On 3 and 4, I've given you probability problems, and I want you to work through these, and I want you to show all of your work. Tomorrow when you get to class, I'm going to be asking 4 different people to show and explain, and the big word there is explain, their answers to the class. Make sure that we all got it. So be prepared for tomorrow to talk about these 4 questions.